This week we are starting a new unit on microbiology. We're going to do 2 weeks of this chapter, chapter 17, fermentation, and then we'll do 2 weeks focusing on food safety. So the first 2 weeks we'll be focusing more on the beneficial organisms plus a general overview of microorganisms, and then in the second 2 weeks we'll be focusing on pathogens and potentially spoilage organisms. So, moving on here. For our first part of chapter 17, we're actually not going to get into fermentation until next week. So, you're reading the part of the chapter all the way up until fermentation on page 504. So, what is microbiology? It is the study of living organisms that are too small to be seen by the unaided human eye, i.e., you need a microscope. So microorganisms, of course, then are living organisms that are only visible through a microscope. We can categorize microorganisms very broadly into beneficial microorganisms. These are the microorganisms that help us with digestion, or microorganisms that we use to make foods, as in fermentation. Then there are spoilage organisms. These are organisms that make our food unpalatable to eat, but not necessarily unsafe. It's just more of a yuck factor. And then there are the pathogens, and these are organisms that, when it comes to food science, make our food unsafe to eat and will make us ill. Now, if we're not talking about food science, pathogens are any organisms that will make you ill. So there's lots of different ways we can categorize the microorganisms. So those three that I just went over are just very broad, the good, the bad, and the really ugly, right? Those aren't really an official categorization, but the three categories that are important to food are bacteria, yeasts, and molds. Viruses can affect foods, like norovirus is really the main one, but we're just going to focus on these three: bacteria, yeasts, and molds. The bacteria are from the Monera kingdom, and yeasts and molds are from the fungi kingdom. So bacteria, overview of these organisms: they are single-celled. They multiply through division, so they just divide and double each, and that is how they reproduce. They have rigid walls and no nucleus. There's no nucleus inside these cells. They are filled with cytoplasm, and that cytoplasm is where all of the metabolism of the bacteria takes place, and within that metabolism is the DNA of the bacteria. And when that bacterium cell divides, then both pieces have all the DNA that the original cell had. And the rate of growth, really just like any of these microorganisms that we're going to go through today, depends on the environment. And that includes the temperature, the oxygen, pH, food supply. And how they grow is the cell will increase in size, and then it will divide in half. And then you have a doubling effect, and there are two bacteria where there was originally only one. A lot of this in this lecture, if you remember it from biology, should be a lot of review. So we can classify bacteria in several ways. Okay, there are three major ways that we're going to go over today. The first is by their shape. So we have the bacilli, which are rod-shaped bacteria, the cocci, which are sphere-shaped, and spirilla. These also can be called spirochetes, but our book is going to go by spirilla, so that's what we're going to stick with. Both are correct, and they are spiral shaped. They're squiggly. A second way that we can classify bacteria is based on their cell wall structure. So the first group has a thin peptidoglycan layer. What does that mean? Peptido. So you can kind of use the root words in here, and you've learned some of this here. Peptido is going to refer to protein, right? Peptides. Glycan is going to refer to a carbohydrate, okay? So it's a protein carbohydrate combination molecule, okay? Plus an outer membrane. So this layer is thin, and it does have an outer membrane also. These bacteria are gram-negative, whereas the bacteria that are have a thick peptidoglycan, which does not have an outer membrane surrounding it, is gram-positive. Now the gram refers to the name of the person who developed the stain that is used to test for these two different types of bacteria. The person was Hans Christian Gram, and he was a Danish scientist who developed this stain in 1884. And so, to give you a better idea of what we're looking at, so this is the outer layer of the bacteria cell. Okay, we have the cytoplasmic membrane, and then outside of that, so as we go up in these pictures, we're going more exterior of the cell. The gram-negative has just a very thin peptidoglycan layer, whereas the gram-positive has a peptidoglycan that is several layers thick. And then the gram-negative has an additional outer membrane that is similar to the cytoplasmic membrane that is surrounding it. Okay. So when this gram stain is applied to either of these bacteria, we get different results. Gram-negative bacteria will be stained a pink color, and that's because the stain is not fully penetrating. It reacts with the peptidoglycan. There's much less peptidoglycan, and it's harder for it to penetrate through the outer membrane. Whereas the gram-positive, when we apply the gram stain, is dark purple in color. And we will see this in at least one of our labs that we are doing this semester. The third way we can classify bacteria, so far we've had shape, and then based on their gram staining, is based on their oxygen needs. So aerobic bacteria, they need oxygen to grow. Without oxygen, they do not grow. The next type is anaerobic, and these bacteria, an means without, and then aerobic is referring to the oxygen. So they need no oxygen to grow. They only grow in environments without oxygen. And then finally, facultative means it can function in either environment, with or without oxygen. Now, often there is a preference, but they can function with or without oxygen. So what this figure here is showing is a visual representation of an experiment that if you were to put these different types of bacteria in a test tube that had a growth medium in it, where they would end up. So obligate aerobes, they would end up at the top, as close to the top as they can be, because they need that oxygen to grow, and so they're going to be where there are small amounts of oxygen that are penetrating that growth medium. The obligate anaerobes, you will end up finding them at the very bottom of the test tube. They are trying to get away from the oxygen, and they'll be as far from the oxygen as it can be. So you think about any like a test tube like this, you're going to have some penetration of oxygen down into this at some level, okay? But at the bottom, there's obviously going to be the least. Now, I just want you to know facultative means that they can be with or without, but just FYI, there's different variations of these facultative. And so you could put them in either the C or D category. And so facultative anaerobes, when we call them that, it means they prefer oxygen, but they do just fine without. So you'll find more of them up at the top, but they do just fine without, and so you'll find them fairly evenly distributed in the layers below where there is high oxygen within the test tube. Aerotolerant anaerobes, they do fine with, without, anywhere they are. Okay, and so they're evenly distributed throughout the test tube. There is a third category or fourth category here. So I'm putting these together as facultative. But this fourth category, or fifth in this image, is microaerophiles. And you don't need to know this one, but if you think about what the word roots are, micro means very small amount, and then aerophiles would mean the aero would be oxygen, and phile means loving. That's Greek. And so they love very small amounts of oxygen. So they're not going to be all the way at the top where there's lots of oxygen, but they will be near the top in an area where there is a little bit of oxygen, but not a lot, and they will cluster there. Okay, so I have the bacteria and from the Monera kingdom. Now we're going to talk about the fungi kingdom, and then I will break that down into the molds and yeasts. So fungi are aerobic. They prefer environments with oxygen. All fungi produce spores for reproduction, and they do not produce chlorophyll. The reason this is important is because fungi used to be classified under the plant kingdom, and this is one of the distinguishing characteristics that led to them being classified into their own kingdom, is that all the plants produce chlorophyll and fungi do not. Some fungi are single-celled, but some are multicellular. So think about a mushroom, that's obviously multicellular, but a lot of our molds and yeasts are single-cellular. And fungi, unlike bacteria, their cells do contain a nucleus. So in this image here, you have the nucleus and all of the fun organelles that we've learned about before in biology. So the basic structure of fungi are filaments called hyphae. Okay, so you have the cells and they form filaments called hyphae. And hyphae are they have cells or chains of cells that absorb nutrients. And these cells form a branch network, or can form a branch network, called mycelium. So it's the mycelium, which are several hyphae chains together. When this happens, you have part of the structure, the mycelium structure, that grows into the energy source. so into the bread, for example, and part remains in the air, and that is what's receiving the oxygen and doing the reproduction, producing spores. These mycelium grow in a circular pattern. So if you look at these mold spots, right, they're circular. So a little bit more about spores. Spores are the reproductive cells of the fungi, and they develop in sac or balloon-like structures, which tend to explode, sending spores into the air, making them airborne and making it easy for these fungi to reproduce, landing in another spot, reproducing, and spread. So here's a little visual of this. This is a puffball exploding and spores being sent up into the air. It's a little bit more in depth about molds now. So molds have a mycelium structure that have a fuzzy appearance, right? They can be in a wide range of colors. This particular image shows a wide range of different colored molds. And some produce antibiotics. Generally, our use of molds in foods, or when we find molds in foods, generally they're not necessarily going to be the antibiotic producing kind. And even so, it's not appetizing to just eat the mold, even if you want antibiotics. So the antibiotics that are produced by molds, I think the book says that a lot of them produce antibiotics. It's not necessarily a lot of them. It's only a certain kinds of molds do produce antibiotics, and other kinds of molds can be toxic even. So proceed with caution there. But penicillins, of course, are our most well-known antibiotics that are produced by molds. Another common antibiotic category that's produced by molds is cephalosporins. Okay, so book note, the book says that molds produce basidiocarps, and so basidiocarps is basically the stem, cap, gill structure, and these are produced by fungi that are in a totally different phylum compared to molds, okay? The Basidiomycota phylum, and that includes mushrooms, bracket fungi, which are like these fungi that you see growing on trees, and puffballs. So totally different group of fungi produce basidiocarps, and the book is wrong when it tells you that some molds produce basidiocarps, because it's a different category. Okay, so now yeasts. So yeasts are fungi that are single-celled, such as this. This is what this looks like under a microscope. They reproduce by budding, and they form, and how they do that, they form buds that swell, and then those buds separate into duplicate cells or chains of cells. So here's a yeast cell, here's the bud. So this bud will swell, it will have its own nucleus, and it will separate and produce that way. Yeast, I think the book says something about them producing mycelium. Some, not very many, and it's not common that yeast actually produce mycelium. So another grain of salt with the book there. Yeast produce ethanol through fermentation, and so you give the yeast a glucose source, and they will use that to produce ethanol and carbon dioxide also. Now this figure shows ethanol plus CO2 in beer, ethanol in wine, and just carbon dioxide in bread. It's actually producing ethanol and carbon dioxide in all three, but in wine the CO2 is removed, and in bread the ethanol is baked out, and the CO2 also comes out too during the cooking process, but it is used to make the structure of the airy structure of bread, right? This image, I'm not going to go through here without showing you this. So this is actually ham. And so if you see these spots on ham like this is yeast growing. And so they grow in small little circular clusters. And so it's very small little dots, raised dots that are like a white, off-white in appearance. If you see that, it's yeast, and yeast does like ham. If this was in a package and it's like sealed and you opened it up, like the very first time you open it up and just like get a whiff of it, it would actually smell yeasty. And like I've had ham where I smelled it and I was like, well, this smells like a barroom floor, like the chicken or something, the Dixie chicken. It smells like old stale beer. And so just wanted to mention that. It's not super uncommon, so if you ever see that, just know that it is yeast. It's not, wouldn't hurt you, but it's not really very appetizing. Okay, so our common characteristics of microbes in general is that they grow rapidly. They can be used to enhance or add to the nutritional value of foods. Because they grow rapidly, they are full of nutrients. They are organisms, right, that are made of the same basic chemicals as us, right? As carbohydrates, proteins, lipids, okay? And so they can be consumed. Now, a lot of times their byproducts cannot be consumed, and if we consume them, if they're still alive, they can really hurt us. So it depends on the microorganism. But there are some weird movements, kind of like the weird movements that people should eat bugs, that people should eat microorganisms because they are a sustainable way to produce food that uses less land, etc., etc. So technically, yes, but it's kind of, I don't see it ever taking off because there's kind of a yuck factor to it. Now, like I kind of mentioned in this last bullet point, many microorganisms do produce byproducts, right? They produce enzymes, but other waste products that may be positive or negative to foods. We'll talk about enzymes in a little bit and how those are used positively. But of course, many byproducts of microorganisms are negative to foods. For example, there's one microorganism that's a spoilage microorganism that really likes to live in lunch meats. And sometimes I've seen it in bacon too. And so if it has grown in your refrigerator, in this lunch meat or your bacon, and you get it out, it's been there for a while, and you pull apart this lunch meat, or hot dogs, it likes to grow in hot dogs also. So you pull this hot dog out, or you pull apart this lunch meat, and you see the slime that is extending from one piece to another. Well, that's one of the byproducts of that microorganism. Now, it's a spoilage microorganism. That slime would not hurt you, but it's certainly gross, and you're probably not going to want to eat it. So that's an example of a byproduct that's negative to foods. Of course, there's other organisms that produce toxins that will actually make you physically ill. And so we have to balance microorganisms, which ones are good and which are bad, and we can use the good ones to our advantage, but we need to do things to avoid the bad ones. The microbes do have the ability to go dormant in response to poor environment, so they don't just die when you subject them to an environment that they don't like. So you can dehydrate them and They don't have water needed to grow, but typically that doesn't kill them necessarily. And instead, they will go dormant and be in a state where they just don't need the water. And they'll stay that way until they get rehydrated, and then they can take off again and start growing. But in the dormant state, of course, they do not grow, they do not reproduce. And so they are, if it's a spoilage microorganism or pathogen, they are less likely to create, or they will not create further negative effects than what is already there. But if they are a good microorganism, then in that poor environment, it can be a good thing to stop their growth. If you've reached to the point where, okay, this is the product I want, now I'm going to stop their growth, right? Or you can use it to your advantage with using starters or dried products that you then bring in and make your foods. So, for example, dried yeast. This is a perfect example of this. When the yeast is dried, that yeast has gone dormant, right? It's not active in this dried state, but as soon as we rehydrate it and give it warmth, it becomes active again. And we give it nutrients, it becomes active again and does the things that we want. Okay, we can also dry bacterial cultures in this way. Often these are freeze dried, and that kind of provides the maximum drying, yet not drying so harshly that you kill them. Okay, because they can be killed, right? Heat kills microorganisms. If the environment is too bad, like they will slowly die off. But freeze drying is kind of a gentle way to dry these. So these freeze dried cultures, they can be used to make starters for food production. So these are a couple examples here. You have this freeze dried culture that is this specific bacteria that is used to make a certain kind of cheese. You can purchase it online and add it to your cheese making process and make the kind of cheese that you want. Same thing for yogurt. Okay. Another example is probiotics. So if you've ever taken or seen pills that are probiotics, well, they're freeze-dried bacteria that have been put into this little pill or powder or whatever it is that you're adding to your food. And then once it gets into your digestive system and is rehydrated and it sees all the nutrients in your digestive system, it comes out of dormancy and is active and able to give its beneficial effects. So the starter, for clarification, the starter is any substance that's containing a microorganism that's added to food to bring about a desired flavor, texture, color change. We have starters have cheese and yogurt examples. There's starters for pickles. There's starters for dry sausage, so like salami and pepperoni. There's starters for beverages, whether it's so like kombucha has microorganism in it. Also, of course, you could say dried yeast is a starter for making alcoholic beverages also. So here's yet another way that we can kind of classify the microbes. This is a loose classification, not the main classifications that we've gone before, but different microorganisms consume different food sources. And so some microorganisms consume mostly sugars and starches, and they're going to produce, let's back up a second, they're going to produce enzymes to digest whatever their food source is. So think about how small a bacteria is. It's not going to be able to consume a whole starch molecule all at once. And so it's going to release enzymes that break down that molecule, and then the smaller molecules that are produced from that are then able to be absorbed through the cell wall and consumed by the microbe. So some enzymes, if their main consumption is starches and sugars, they're going to produce carbohydrates. There's a table on carbohydrates or on carbohydrates on page 500. We've essentially talked about these enzymes before, just not related to bacteria. So amylases, they convert starch to sugar. And so back up a second. So when we're talking about beneficial uses of microbes, we will take advantage of the fact that they produce these enzymes to potentially use them to help us. And we've talked about how we use enzymes in food production before. So just overview here. I'm not going to have it all here on the slide. It does talk about it again in the book. We've talked about it before. So amylases and the carbohydrates, they convert starch to sugar, right? Why? We would use these for baking, for brewing beer, for making syrup. Cellulases, they break down cellulose and make it into sugar that can be fermented, or can be used, or the cellulases can be used to remove cellulose from fruit juices, or which would clear them up, right? The clarifying effect. Sucrases, they break down sugars, either for use in fermentation or to have higher fructose and the sweetness from that. Invertases, they convert sucrose to glucose and fructose. They can prevent crystallization and can be used to make artificial honey. And then pectinases, they break down pectin, and we've talked about how that can be used to clarify juices also, and can also allow more juice to be extracted from fruits. So those are examples of the carbohydrates and how we would use those carbohydrates that are produced by the microbes for food production. So microbes that consume primarily protein, they produce proteases, enzymes that break down protein, and we might use those for different reasons. If those bacteria that do consume protein, we give them a special name. We call them proteolytic bacteria. Bacteria that consume mostly lipids, they are going to produce lipases, which of course will break down lipids, and those bacteria we call lipolytic bacteria. And there's another type of microbe that are called halophilic. Most microbes have lower tolerances for salt, and salt will slow or stop their growth because it dehydrates them. But there are some that are more adapted to salty environments, and they're called halophilic. They don't necessarily produce a special enzyme, but I just included this with the classification based on food source. Now, like I said before, microbes need the same basic chemical nutrients as us to survive, right? They are made of carbon, hydrogen, oxygen, and nitrogen. I might have this on the next slide. Maybe not. Okay. And so their food sources need to include those, right? Because they need to be able to make protein. They need to be able to make their own carbohydrate. They need to be able to make their own lipids. And so in general, they're going to consume these things, right? Sugar, starches, proteins, lipids, but some more than others. Now, all bacteria are going to need to consume protein to some extent because they need nitrogen. They need nitrogen source to produce their own protein and their own DNA. But they might consume more carbohydrates, and this is the main, more carbohydrates, and so carbohydrates are the main enzyme they produce, but they just need enough protein to get by, and so they're not focused on consuming protein. Most most microorganisms, carbohydrates are the main focus and not protein or lipids. Okay, so how are microbes scientifically named? Okay, we refer to all of the microbes using their genus and their species. Okay, so for example, Lactobacillus acidophilus is the one used in the book, and we'll just go through that example. So now the first time you use the name of a bacteria, you spell it all the way out. So if you're writing about a bacteria, writing a paper or something, the first time you use it, you spell it all the way out. And that's because this, well, I'll go into that in a second. Okay, so you also always italicize the full name of the microbe when you type it. The genus is always capitalized. It's not just capitalized here because it's the beginning of my bullet point. The genus, this Lactobacillus, is always capitalized. Now so go back. The first time you use the name of the bacteria, you always spell it all the way out. But then afterwards, you can use the initial for the genus. Okay, so after the first time I've written about Lactobacillus acidophilus, well, then after that, I can say L, acidophilus. Now, why do I spell it all the way out the first time? Well, that's because there's other genera that start with L, right? And so if I've spelled it out once, I've told my reader, okay, it's this genus and species. And afterwards, I'm like, okay, we've already told you what genus exactly it is. And so now I'm just going to abbreviate it. Now, unless you had two different genera that were both acidophilus species, then you'd have to spell it out all the time, but that doesn't really happen. Okay, then species is never capitalized. The name also tells us more about the organism because most scientific names are Latin in origin. Knowing some Latin can help us with determining what an organism actually is. So this particular organism, so the lacto, well, this sounds like comes from milk, right? Or lactose. And so we can assume that it probably consumes milk. Bacillus means it's a rod-shaped bacteria, and actually several different genera names indicate that they're rod-shaped. If they end in Bacter, if they end in Monas, if they end in Ella, they're all rod-shaped. If they end in Coccus, it's a cocci bacteria. That one's pretty obvious, right? So then the second part, acidophilus, okay, from that we have acid and phylus loving. So, or actually, I don't think this is loving. So actually, just acidophilus, we know that it is producing an acid. So basic point to get from this is since most genus and species names are derived from Latin, if you know Latin, it can help you more with understanding more about the bacteria just by looking at the name. So there's a use of your years of Latin in BCA. Okay, so now we're going to go into 4 factors that affect microbe growth. And I'm just going to give you one slide with a list of all 4, and then we'll go into each one. So the food supply, so what is the environment that the bacteria is, what food is there, nutrients are there for the bacteria or fungi to consume, what water is available, what is the pH, and the temperature. Now, all of these work in tandem. Okay, so if the food supply is good and the pH is good and the temperature is good, but the water availability is too low, the bacteria or whatever other microorganism will not grow. If you have a good food supply and water and pH, but the temperature is too low or too high, the bacteria will not grow. And if it's too high, you've just killed it. So we have to be able to optimize all of these things if we are wanting the bacteria to grow. Now, if we're not wanting the microbes to grow, then we want to make them as negative as we can. Now, we can't always make all of them or even one of them all the way negative because we might not be able to consume that product, right? Like temperature. Well, we can't serve everything frozen. pH. We can't make everything acidic, so acidic that the bacteria can't survive, or basic. Like we might destroy the food properties if we do that. Water. Well, some food we want it to be juicy, right? So it's going to have high water availability. And generally, our food supply is very good for microorganisms, right? They like to eat the same things we do. But what we can do, and we'll get into this more in food safety, but we can make each one of these just a little bit not great for bacteria. And then the sum of all that makes it so bad the bacteria do not grow. So if our goal is growth, we need to make sure that all of these factors are ideal. If our goal is stopping growth, we can often make several of them a little bit not ideal, and it adds up to a very not ideal situation for the bacteria if they don't grow, or whatever other microorganism. Okay, so the first one is food supply, the first factor that affects microbe growth. So food supply is the environment that the microbe is in. It will consume whatever it is in contact with, that it has the enzymes that can help break down. Okay, this is what I was talking about before. I was like, I think I had this on another slide. Yeah. So the environment must supply carbon, hydrogen, oxygen, and nitrogen. Okay. So most often then the food supply is going to need to be from at least one of the molecules that provide these main nutrients, right? Carbohydrates, lipids, or proteins. And like I said before, only proteins have the nitrogen, so that means all microbes need protein at some level, but some more than others, okay? Like bread has protein in it, right? And it can have enough for many microorganisms. So that food supply that's needed depends on the enzymes produced by the microbe. What can it actually digest? So if the microbe does not have, or the food supply, sorry, if the microbe doesn't have carbohydrates, for example, or good carbohydrates that would actually break down bread, well, it can't break down bread, right? If it doesn't have lipases, well, it can't break down lipids. And so the microbe needs to be able to produce the enzymes that that would break down the food supply that is around it in order for it to be a good food supply for those microbes. Okay. The microbes do also need some level of vitamins and minerals. Just like in our bodies, vitamins and minerals act as coenzymes and are necessary for biological functions of microbes as well. So microbes also need available water, and so with that we talk about water activity. So the simple definition of water activity, which is abbreviated with an A and a subscript W, so it's activity of water. So the simple definition is that it's a measure of how much water is available for microorganisms to grow in the material that you are measuring. And the more scientific, precise definition of how that measurement is achieved is that the ratio of the vapor pressure of the material to the vapor pressure of the water. So vapor pressure of the material, and they divide it by the vapor pressure of water. So water, pure water would have a water activity of 1, okay? And all water activities of anything else is going to be below 1. So food can basically be considered completely shelf stable at water activities of 0.7 or lower. There aren't, like with that 0.7 or lower, we're talking like crackers, okay? That kind of thing. So what water activity does each microbe need to survive? They do vary based on the type of microorganism. So bacteria need a water activity that is, I'm going to adjust this a little bit, but we're just going to say over 0.83. But spoilage bacteria need a water activity above 0.9. So you can stop the growth of most spoilage bacteria if you can get that water activity below 0.9. Okay, and so there are spoilage bacteria are quite prevalent in the environment, and a lot of other bacteria are not. Okay, so spoilage bacteria are usually our main concerns. So then yeasts, they prefer 0.87 to 0.94, so they grow at about the same water activity as bacteria. Molds, on the other hand, can grow at water activity that is substantially lower than bacteria or yeasts. Now, this is preferred. There's usually a range outside of that where the microbe can grow, but it's not optimal. It's not its fastest growth. I did want to go through some food examples just to kind of show you. The percent water that is in a food is not indicative of what the water activity is going to be. And the reason for that is there are other things besides just the amount of water in a food that affect how much water is available for bacteria or for molds or for yeast to grow. So sugars in a food decrease the water activity. Salts in a food decrease the water activity. All the other things in a food can affect the water activity. It's not just about the water. Sugar and salt are some of the main factors that will decrease water activity. Okay, so milk is 87% water. It's the water there that is available for microorganisms, 0.99. All right, 99% of that water is available for the microorganisms to grow. That's kind of a way you can think about the water activity. So cheddar, which is made from milk, has had a lot of the water content removed, quite a bit, right? Over half of it has been removed. But that water activity isn't much less than milk. It's at 0.95. So 95% of the water that's in cheddar is available for microorganisms to use. Okay, hot dogs. The water content, about 57% on average. We know that there is a big range from our hot dog lab that we did a long time ago, but 57% on average. The water activity is 0.95 on average of hot dogs. Great environment for bacteria to grow. Ketchup, water content is 70%, but it actually has a lower water activity than hot dogs. It has greater moisture, lower water activity, and that's because it has more sugar, especially, in it. Now, it's still a great environment for things to grow, and what stops the growth is what we're going to talk about next, is pH. Maple syrup. Maple syrup only has a water content of 30%. It's very high in sugar, which decreases that water activity substantially down to 0.85. So most bacteria that would spoil maple syrup are not going to grow in that medium. Okay, grape jelly, it has higher water content than maple syrup, but lower water activity, has even more sugar. And so its water activity is 0.77. So bacteria and yeasts are not going to grow in grape jelly. What will grow is mold. And so if you've ever seen mold growing in jelly, that's partly why, because it has a great water activity, a water activity that is suitable for molds, but not suitable for bacteria or yeast. Okay, so now pH, our third factor that affects microbial growth. I'm just going to have this table up here to describe what's going on here. So like water activity, the pH that is necessary for growth is very dependent on what microbe we are talking about. And this table is very general, saying like yeast prefer this and molds prefer this and bacteria prefer this, but in bacteria especially, there's a wide range of things that they prefer based on what actual bacteria it is we're talking about. Bacteria are very diverse, I should say. Okay, so most bacteria, I've adjusted this number from what the book says. I forget what number the book says, but I put 4.6 here. Most bacteria will not grow below pH 4.6. And this is a very important number in food science for food safety reasons. If at all possible, I will say this. So anything that is below pH 4.6, we call it a highly acidic food, and it requires much fewer food safety steps, food safety temperature requirements. It's much more safe just on its own without us adding extra interventions to stop bacterial growth, because most bacteria will not grow below pH 4.6, especially most pathogenic bacteria. So notice yeasts and molds have a lower threshold than bacteria and a lower preferred range than bacteria. Yeasts and molds like to grow in a more acidic environment than bacteria. In general, bacteria prefer a more neutral environment, right? Notice 6.5 to 7.5. We're half a point above and below pH 7, which is perfectly neutral. Now, their maximums, molds just have a very wide range at which they can grow, right? They have the lowest minimum and the highest maximum. Yeasts have a lower maximum than bacteria. But the key point here that I want you to understand is really this minimum on the yeast and molds. They grow much more easily in acidic environments. And so we still have to potentially be concerned or take advantage of that if we are using beneficial bacteria. Whereas our beneficial microbes in our foods. Whereas bacteria, they really just prefer an environment that is pH neutral. Okay, and our last bit, it's not the last slide, I have 2 slides on this, but last factor that affects microbial growth is temperature. So cold can either slow or stop the growth of microbes. Heat can kill microbes. So in general, unless we're talking about a very, very long time. Cold will not kill microbes. They will just go dormant and will come out of dormancy if you thaw the product up. Okay? But it will stop their growth if you get it cold enough. So, but heat will kill the microbes. It affects all of their systems. It causes denaturation of proteins, etc. Much more so than cold does, and so it will kill them. In general, the microbes will usually die at about 5 to 12 degrees Celsius above the temperature at which their maximum growth occurs. So they have a range at which they're okay, and that range is different for different microorganisms, and if you get too far above that range, they die. Pasteurization, then, is the process in which generally liquid is heated until pathogens and some spoilage microorganisms are destroyed. So this is not sterilization. Sterilization kills everything. But pasteurization, the key thing here is that it's heated at such a temperature that all of the pathogens are destroyed. So if we're talking about milk, every pathogen that is known to be potentially present in milk is destroyed at the temperatures at which they heat the milk. Now, this pasteurization process may change the food attributes because it's heat and you're denaturing proteins and affecting enzymes. So you may change the flavor, the texture, etc. of milk. So the people that prefer raw milk, like, it's a real thing. Pasteurization does affect the flavor and texture of the milk. And so it's not just in their heads that the raw milk tastes better for them. It does taste different. Now, I'm going to talk a little bit more about milk pasteurization because for me it's kind of interesting. Most of the milk in the US, the pasteurization process is that the milk goes through these heated tubes. And so it goes through this super hot tube and it's heated, and it stays in this tube that is heated for about 10 to 20 seconds. And it gets that milk to a temperature of 165 degrees just for that time. So it's not very long that it's heated, but this temperature for this time combination is enough to kill any pathogens that are potentially in the milk. And so it is going from the big vat of milk into the superheated tubing, and then it goes directly into tubing that chills it down. Okay, and the goal there is that it's heated for the minimum amount of time possible, or minimum amount of time needed to kill the microorganisms. And then it's immediately cooled so that the damage that can be caused by pasteurization that changes the food attributes is minimized, and it tastes closer to raw milk. It's not perfect, but it's better than what we used to do, where you had a whole vat of milk and they would get that whole vat of milk for up to 145 degrees Fahrenheit for at least 30 minutes. Now, the problem with this vat is it's going to take a long time for it to get to that temperature, and then a long time for it to all get down. So it's much more than this 145 degrees for 30 minutes, because you're also at 135 degrees for 10 minutes, on either side of it, etc. Because it takes time for that milk to heat up and cool down, and so you're actually at high temperatures for a longer time than even what it says, 145 degrees for 145 degrees for 30 minutes. Now, another method of pasteurization that is not used as often, but it is used, is what we call ultra pasteurized milk. And this is heated for just 3 seconds, but it's up to 280 degrees. So same kind of process. It's going through just superheated tubing, like piping, and just for 3 seconds. But this short amount of time at this high temperature actually produces milk that is shelf stable, which means it's not only killed all of the pathogens, it's killed all of the potential spoilage microorganisms also. It is completely free of bacteria that can grow in the milk. And so you can then package that milk into bottles or cartons that can sit on the shelf at room temperature and be perfectly fine. So a lot of the fast food restaurants that sell the little jugs or cartons of milk for like their kids' meals and stuff use this type of milk so that they don't have to worry about refrigerating it and throwing it out if it is past its expiration date, because it has a very long shelf life also. But it definitely does affect the flavor. I cannot drink this stuff. It's disgusting. So I don't ever order milk from fast food restaurants because I'm afraid that it will be this kind of milk and I can't drink it. They've also, the military uses this. My husband, who was deployed to Iraq, said that this was the only kind of milk that they gave them in Iraq because they didn't have to worry about it going bad in the desert. So, okay. So I have one more slide on temperature here. And this is a little bit more information that is in the book, but I want you to know it. Different bacteria or microorganisms, even different microorganisms prefer different temperatures. So some grow at cold temperatures, some prefer body temperatures, some prefer warm temperatures. And knowing your bacteria type and what you need to encourage the growth of or discourage the growth of can be really important. So the first group is psychrophiles. So psychro, all of these are Greek origins for the roots of these words. Psychro means cold, philes means loving. So cold, loving. So these grow best at 0 to 15 degrees Celsius. This is refrigeration temperature. There are actually some bacteria that grow the best at refrigeration temperature. Psychrotrophs are kind of related to psychrophiles. So psychro still means cold, and trophs means nourishment. So they get their nourishment in the cold. And they grow from 4 to 25 degrees Celsius. This is still refrigeration temperature, but it also goes up a little bit higher that they like. Now, all of these can grow a little bit beyond these temperatures, but this is the range that they grow best at. Most of our spoilage organisms are psychotrophic. So specifically the ones that grow cause spoilage of food while it's in your refrigerator. So they come from warmer temperatures, but they thrive just fine at colder temperatures and consume your food and produce their byproducts and yuck factor. Okay. Mesophiles. So meso means intermediate, philes loving, so intermediate loving. So they grow best at these intermediate temperatures from 20 to 45 degrees Celsius. That's this green one here. This is body temperature. Okay, body temperature is 37 degrees Celsius, which is right in this range. Most of our pathogens, the things that can make us sick, are usually mesophiles, and that makes sense because they can grow in our bodies. But they don't grow well at refrigeration temperatures, right? But if they're already there, they're not going to die. Remember, they are still just sitting there, and when we consume them, if they're there, then they'll be in our bodies. Thermophiles means hot loving, and they grow best at temperatures above 50 degrees Celsius. There's actually two categories of these thermophiles, and the hyperthermophiles, which really love hot temperatures, but we're just going to group them all together in one group. thermophiles and hypothermophiles together. Anything above 50 degrees Celsius is a thermophile. So there are some bacteria that not only tolerate heat, but love it. Most of them don't grow beyond boiling temperature, though, and will in fact die if you get it to boiling. With some exceptions, which we'll talk about in food safety. So that is our first part of chapter 17. The chapter's on fermentation, but really this first part is just a big overview of microorganisms in general, and hopefully plenty of this was review from biology, so it's not too much information overload for you.